Hey folks, and welcome to another edition of Zog Science. Today we're going to be talking about immunity and the immune system. So, who has an immune system? Well, all animals and plants have an innate immune system, and also some fungi and bacteria have kind of a proto-immune system as well. We'll talk about them at the end. However, only vertebrates have acquired immunity, and innate immunity is kind of made up of two things. You got barrier defenses and internal defenses, and we'll get into what those are. Acquired immunity is a little different. It is going to be recognition of specifics and traits that have to do with particular pathogens, and it includes the humoral response and the cell-mediated response. So let's get into it. The vertebrate immune system consists of two main types of things. You've got the lymphatic system, which drains the body of toxins, and you have leukocytes, or white blood cells. Remember, we talked about those when we talked about the circulatory system, and those are going to be patrolling the circulatory system. Now, the innate immunity is a non-specific immune reaction or immune system reaction, and again, it's made up of external and internal factors. The external factors are skin, mucus, and ciliated epithelium, and the internal factors are the phagocytes, inflammation, and toll-like receptors. So skin, which is one of our external factors, is a waterproof barrier, and it just helps to push things out, or not necessarily push things out, but keep things out. We have a lot of commensalistic bacterial species that live on our skin. They do not harm us, and they take up niches that prevent other, pathogenic bacteria from growing on our skin and potentially getting inside of us, which is good. We also have our sweat makes the skin acidic, keeps it at a pH around 4 or so, and that is a very harsh environment for a lot of pathogenic bacteria, and so it makes it difficult for them to live. So it's actually a good thing that you sweat. You also have oil glands that contribute to making it an environment that's not very good for a lot of pathogenic bacteria. Then we've got mucus. Mucus is very important. It lines surfaces not protected by the skin, so you can think about, your respiratory tract, your digestive tract, things like that. It is a physical barrier, so it actually, physically prevents bacteria and other organisms from crossing it. It also, however, is a chemical barrier because it contains lysozymes, which help break down bacterial cell walls. So that's really good. It actually kills bacteria. Now, the reason why the mucus is important is that we also have ciliated epithelium. Epithelium, remember, those are lining cells. So ciliated epithelium are epithelial cells that have cilia. You can kind of see what those look like right here. And so what happens is that mucus traps a pathogen, and then cilia moves that mucus into the digestive system. So you have mucus that is being produced in your lungs, that is moved up into your throat and then down into your digestive system, or mucus in your sinuses that is pushed down into your digestive system. And once it's in there, the digestive system destroys the pathogen, making it safe. So it's actually a good thing that you have all this mucus and that it does actually get down into your stomach. You are built for mucus to go down into your stomach. So that's a good thing. Next, we have some of our internal factors, such as phagocytes. Phagocytes are types of white blood cells. They are non-specific, so they're just going to engulf any unrecognized cell. And then digested pieces can be presented to the acquired immune system for a further response, and we'll talk about that once we get into the acquired immune system. This is kind of how the process of phagocytosis goes. Here's some microbes. They get surrounded and engulfed. They are then combined with the lysosomes, which contain the enzymes. The enzymes break down the microbes, and then those can be released. This is a scanning electron microscope of a phagocyte engulfing some sort of pathogen. Most likely, it looks like a bacteria. We haven't done a video within a video in a little while, so I've got a quick video that kind of shows you how phagocytes.
Microorganisms that cause disease are called pathogens. When pathogens enter the body, white blood cells called phagocytes pass through the walls of the blood vessels and into the surrounding tissue. There they can ingest the pathogens and pathogens' toxins, absorbing and digesting them. Phagocytes can also destroy pathogens by releasing enzymes. Having absorbed a pathogen, the phagocytes may also send out chemical messages to call in other white blood cells. Microorganisms that cause disease are called pathogens.
Now, the way that phagocytes actually will begin to identify whether or not an organism is a pathogen or some sort of non-self cell is that it has these toll-like receptors. And these TLRs are present on the cell membranes, and they are able to basically bind with or detect foreign molecules, such as things like a flagellum or lipopolysaccharides. The reason why they are specific for those things is that pretty much humans, except for sperm cells, don't have flagella. We have cilia, and we do not have lipopolysaccharides. Those are things that are very common to bacteria. And so what happens is that when a TLR senses those, or maybe senses some DNA, it is going to then help initiate the phagocyte response, which is either to help engulf the pathogen, or in order to initiate the inflammatory response, which is the final part of the innate response. So what happens is that we have these mast cells and other macrophages, and when they engulf a pathogen or encounter a pathogen, they are going to begin releasing cytokines. And these cytokines are chemical signals that are going to do a variety of things. Some of them cause capillaries to dilate. Now, once the capillaries start to dilate, fluids and other phagocytes, different types of phagocytes, such as neutrophils, are going to be attracted, as well as some peptides. So there are some proteins that have antimicrobial properties. Those will kind of be released into the injured area, and the swelling is going to begin to occur because now you have all this fluid and things that are getting there. So swelling is actually a good thing. It is a healthy response in this kind of situation because it promotes healing. Eventually, the neutrophils are going to digest all the pathogens, and healing is going to begin to occur. The swelling will retreat as the number of cytokines released decreases. All right, so that's it for the innate immune system. Now we're going to talk about adaptive immunity, which is probably the immune system response that you've heard the most about in your life. It is the one that most people tend to talk about. Let me make this full screen again. All right, so the adaptive immune system is a specific immune system, and it is specific to particular antigens. And there are two different types. We have the humoral response, and we have the cell-mediated response. The humoral response deals with extracellular pathogens and involves B cells, while the cell-mediated response deals with intracellular pathogens and cancer, and it is going to involve cytotoxic T cells. So we're going to begin with the humoral response. But before we get to that specifically, we need to talk about kind of overall what's going on with the adaptive immune response. The cells that are involved are the B cells and the T cells, which are more generally known as lymphocytes. B cells mature in the bone marrow, and the T cells mature in the thymus, which is located just above the heart in the thoracic cavity. So you can see pretty easily where they got their names from. Now, the specificity of the adaptive immune response comes from the fact that B and T cells have antigen receptors that are found on the outsides of them. Now, if you look at the different antigen receptors, you'll notice that they have a V region and then a C region. The C regions are the constant regions. You can find them on both the chains found on B cells and on T cells. And those are always the same. They're what help anchor the receptors into the cell membranes. But then on the ends, you'll notice that you have these variable chains. And the variable chains or the variable regions will change based on whatever combination of genetics that particular cell has. And these are a bunch of introns and exons that can be excised out to produce different combinations. And we can end up with over 3.5 times 10 to the 6 numbers of possible receptors that you can have, which is excellent because that means that you have such a wide variety of receptors that in all likelihood, one of the receptors is going to be able to bind with the antigens found on the outsides of a particular pathogen or disease-causing agent. So it's very good that we have these binding sites, and that the fact that they can be so variable means that it basically allows the immune system to do what it's supposed to do. So in a first encounter, helper T cells are going to be the first ones to respond. So what happens is that you have some sort of phagocyte, could be a neutrophil, could be a macrophage. It is going to ingest a antigen-presenting cell. It is then going to use a class 2 MHC molecule in order to present the antigen. So the class 2 MHC molecule binds to the antigen. It then presents it, and the helper T cell is going to come along, and it is going to recognize the antigen. So it has the proper T cell receptor to match up with that antigen, and it is going to say, okay, I recognize this. I'm going to initiate an immune response. And so it's going to begin to release, proteins, signaling molecules that are going to activate B cells, and they are going to activate cytotoxic T cells. So let's see what happens when the B cells become activated. And what is going to happen in the humoral response is that we are going to we're going to eventually get to the production of antibodies. And antibodies are soluble antigen receptors, so you can imagine what they're going to do. They're going to bind to the antigen wherever they find it. So what happens is that you have a B cell. That B cell has bound to the antigen, and it is going to begin to present using a class 2 MHC molecule that antigen. A helper T cell that has also encountered that same antigen and is now activated, it will bind with that particular B cell, okay? So once it binds with that particular B cell, it's going to release cytokines that are going to tell that B cell, hey, you need to activate, okay? You have an antigen. I am confirming the fact that you have this antigen. Now you need to produce 2 different types of cells. You're going to produce some plasma cells, and you're going to produce some memory B cells. And we'll talk about what each of those does in just a minute, right? So again, here's just kind of what is going to happen with that humoral response. Phagocytes present antigens to the helper T cell. The helper T cell becomes activated. It will then activate B cells that are displaying the same antigen. The B cells then proliferate, and they are going to turn into 2 different types of cells. Number 1 is the plasma cells. These plasma cells produce antibodies specific to the antigen, so they're just going to start spewing out all of these antibodies. Those antibodies then go around and they start binding to the antigen. And then we also are going to spawn memory B cells. The memory B cells are very important because they are going to be sticking around for a long period of time. Those memory B cells keep on replicating, and they're going to constantly be remembering the fact that we have seen this antigen before, and if that antigen ever comes along again, the memory B cells will turn into plasma cells and just start releasing antibodies. They can skip the step of the phagocyte having to activate the helper T cell, and then the helper T cell activating the B cell. So antibodies are also known as immunoglobulin, or IG for short, and there are 3 different things that they can do. They can either do viral neutralization, and that's where when the antibody binds to the virus, it prevents the virus from being able to infect cells, which is obviously really good. They can do opsonization, which is where when the antibody binds to the bacteria, it is going to induce a macrophage to come along and ingest the bacteria, which is good. It can also activate a complement system where the antibodies bind to the antigen, which is on an actual foreign cell. So this could be a pretty large cell, maybe some sort of protist, something like that. And the complement proteins are going to come along, and it's actually going to eventually form a pore in the cell membrane of the cell and lead to the cell being destroyed because water and ions and other things are going to be able to pour into the cell. So antibodies are pretty good at their job. Now, how B cells proliferate, okay, just as a quick aside, all B and T cells are screened for self-reactivity. So we said that the B cells just kind of, they have the ability to recognize certain antigens, right, based on what receptors they have. Well, you don't want those receptors to be recognizing yourself, right, because then all of a sudden you're going to begin to attack your own body, right? And we call those autoimmune disorders, and we'll talk about them at the end. Same thing that happens for T cells. So they're, first of all, they're considered for only only ones that don't attack the self are allowed to proliferate, right? Then once an antigen is produced, we go through clonal selection. Clonal selection is where, okay, these antigens are present, so we need these cells that are able to recognize those antigens. We want those to begin to replicate, and that's where we get the different types of B cells, the memory cells and the effector or the plasma cells is another way to say that. And then we said that we had those memory B cells. Again, those memory B cells, they improve response to a second time that we see the specific antigen. Someone who is considered to be immune, right, has memory B cells or also memory T cells, and for a particular pathogen. So what we can see here is that we've got exposure to an antigen. About 14 days later, we peak out at our response with the number of antibodies for that pathogen. If we are exposed to it again later on, notice that the response is much greater, and it also happens much faster. And that's what that secondary immune response is, and that is all due to the fact that you have the memory B and T cells that are present, and they help to combat diseases, combat pathogens much quicker, which is the whole point of a vaccine. All right, so the cell-mediated response is T-cell mediated. It's controlled by T cells, and it results in activated T cells that destroy infected cells. Basically what happens is that we have a helper T cell that is going to activate the cytotoxic T cells and move on from there. So here's how the cell-mediated response works. The T cells are going to go through the same activation process as before. Helper T cells can either help activate the cytotoxic T cells, or they can help activate the memory T cells, which will then turn into can eventually turn into the cytotoxic T cells. Or you can also just have an antigen-presenting cell activate the cytotoxic T cell automatically on its own. Again, memory T cells are going to do the same thing as memory B cells. They're going to remember the antigen for a secondary response. The cytotoxic T cells are going to kill the cells expressing the particular antigen. So how that works is that the cytotoxic T cell is going to recognize a class 1 MHC molecule, not a class 2, because the class 2 are only presented by phagocytes. So it's going to recognize a class 1 MHC molecule, which is put out by other cells. It is then going to release chemicals, and those chemicals are going to create pores. The chemicals are perforin and granzymes, and those are going to form pores in the cell, which is then going to destroy the cell. It's going to kill it off because now it has got all these pores in it. It's going to be leaking fluids all over the place, all that kind of good stuff. All right, so we've got allergies. Allergies are an immune response to non-pathogenic antigens, and this is going to eventually lead to an inflammatory response. The reason why is that the immunoglobulin is going to trigger your mast cells to release antihistamines. I'm sorry, not antihistamines, histamines. You can take antihistamines to combat this problem. And that inflammatory response is what you feel when you're having an allergic reaction. Now, obviously, you can have a much larger, allergic reaction called anaphylactic shock, which is where your entire body can shut down because you just have gone through way too much inflammation, and that is very bad and can lead to death, which is why people might carry an EpiPen, which has epinephrine in it. That epinephrine is able to combat this this systemic swelling, the systemic inflammation due to the release of all of these histamines. You can also take things like antihistamines, such as Claritin or other things like that, in order to help combat allergic reactions. And now you can also have autoimmune diseases, and this is where your immune system responds to your own cells, so it actually attacks your own cells. There are lots of different kinds. You've got a whole list of them down here. One of them, lupus, right, is always what they think it could be in-house, but it never actually is. And another example would be arthritis. And so in arthritis, your immune system is actually attacking your joints, which is not good because your joints become inflamed. They can become distorted and things like that. You can take immunosuppressive drugs, which will actually, curb your levels of white blood cells. Not always the best, though, because then you're opening yourself up to other infections, which is what happens when you get AIDS. So AIDS is an infection of the helper T cells by the HIV virus. Because it is invading your helper T cells, it is difficult for your body to mount an immune response against the HIV virus. It is going to lead to a decrease in the helper T cell population. And as the helper T cell population decreases, you become more susceptible to other random infections, like maybe something like the common cold or something like pneumonia, right? Your body is not able to properly fight against it because its helper T cell concentration is too low. And so eventually you get AIDS, which is the acquired immunodeficiency syndrome. And that is going to get to the point where you can't fight any infections, and eventually you can die from something like the common cold. Antiviral drugs can help, and there are a variety of treatments available. However, they are pretty expensive, and you basically have to be on drugs for the rest of your life. So not the best. Now, we mentioned the idea of vaccination earlier. Vaccines, the whole point of a vaccine is to expose the immune system to the antigen without exposure to the pathogen, right? So you want to expose the immune system to the antigen to elicit the creation of memory cells. That way, the next time you encounter the antigen, you are able to fight it off properly. However, you don't actually want to expose people to the pathogen so that they don't actually get sick. That would be totally counterproductive. So the way that you do that is you maybe, isolate the antigen. You maybe, take, just like if it's a virus, you can take just the husk of the virus, so the protein coating on the outside, something like that. The problem with this, obviously, is that antigens can change, right? So evolution is going to be occurring. They're going to change their antigens. And so not all vaccines confer lifetime immunity. For example, the flu vaccines, right? We have to get a new one every single year. Now, there's been a lot of talk about the measles, mumps, right, making comebacks. The reason why they're making comebacks is because people haven't been getting vaccines, okay? This is so silly, okay? There was an article, research done maybe 10 years ago that has been totally debunked that said that there could be a link between the measles, mumps, rubella, the MMR vaccine, and autism. That is totally not the case. Vaccines do not cause autism. Please, 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 please get vaccinated. Make sure your children get vaccinated whenever you have children. This is concept of herd immunity. Okay, you want to make sure that you're vaccinated. The more people that are vaccinated, it protects everybody else. When you get people who are not vaccinated, as you get more and more people who are not vaccinated, then that means it's much more likely for individuals, especially those who are very, very young or those who are very, very old, to die from these simple things that we could totally have eradicated if we continued with vaccination. So please be sure to get vaccinated. We've mentioned this a little bit, but other organisms do have kind of immune systems. Bacteria, we've talked about this before, use restriction enzymes to counteract bacteriophages. Obviously, the bacteriophages are going to be undergoing evolution and have, evolved ways to counteract that. Fungi produce chemicals, so things like penicillin is actually produced by a fungi, and so they produce chemicals that can be antibiotics. Plants have innate immunity, and they do have some acquired resistance as well, although obviously we're not going to get into any sort of details with that. So thank you for watching, and I'll see you next time.